Hello, my name is Dr. Karl Walker. I'm an assistant professor in the Department of Mathematics and Computer Science. I do research in the area of bioinformatics. Now, bioinformatics is a new field that's at the precipice of biology and computer science. So basically, it's a combination of both biology and computer science. And what we do is we analyze a lot of the biological data from different biological experiments. And those experiments can be from the main three molecules in molecular biology, which is DNA, RNA, and protein. My research specifically deals with protein. And so I received funding from the Arkansas Embry of a little over $50,000 to conduct research on the sequences related to different structural transitions in proteins. As we know, proteins are composed of amino acids. And the unique arrangements of amino acids constitute a specific protein. And by trying to figure out how certain patterns of amino acids relate to certain structures, we can try to predict the structure of a protein from its amino acid sequence. So my research mainly deals with this. And the way that we do this is that we look at data that's already been predetermined from different experiments. And most of these experiments are X-ray crystallography or NMR experiments. And we get this data, we load it into databases, and we look at the structures, different structural relationships in the sequences compared to the structure, the three-dimensional structures. And specifically, this research, we look at secondary structures and divergent omega dihedroangles. Those are the two structures we're interested in as far as this study funded by the Arkansas Embry. And the Arkansas Embry is also part of the National Institutes of Health and the National Institutes of General Medical Sciences. This is an opportunity to increase bioinformatics research at the University of Arkansas at Pine Bluff and to expose students to the entire pipeline from biological experiments to knowledge, the gain of knowledge through these experiments. Because once you have the data, you have to analyze that data in order to mine out the different knowledge that may be contained within that data set. And so students get exposure to working in the lab, attending various conferences and workshops, and also meeting other researchers around the state and around the country. And this helps students to figure out what they like to do and also what area of bioinformatics research or any other field of research that they would like to find themselves working in the future. I have both undergraduate and graduate students working on this project. I have some undergraduate student looking into the sequence patterns related to changes in omega dihedroangles. Now, omega dihedroangles are usually fixed at 180 degrees due to the double bond characteristic of peptide bonds. However, under certain circumstances, this bond is able to break and the omega torsion angles become more flexible. And so we're trying to find out if there's a sequence pattern related to this change in omega dihedroangles. And also have students looking into different secondary structures of proteins. So proteins have an overall three-dimensional shape, but they also have short three-dimensional patterns that develop locally along the peptide chain. And so we're looking at different secondary structures such as alpha helices and beta sheets and trying to figure out sequences related to their transitions. So basically we're looking at the boundaries of these secondary structures and trying to find the code that's related to that change and transition. I'd like to thank Dr. Larry Cornett and Dr. Helen Benesh at the Arkansas Embry for this opportunity to continue this research in protein structure prediction.